Hello, everybody. The world is facing a pandemic in the form of COVID-19, the coronavirus disease. Now, all of us know what exactly are the symptoms of this disease, the causative virus, which is called as the coronavirus, because of which it's called as a coronavirus disease or COVID-19. We also know the precautionary measures that we need to take to prevent getting infected by this virus. I would like to emphasize on a few important details of this virus as a biologist. Now we know what a virus is. A virus is a submicroscopic infectious agent. Rather, you can call it as a particle. It's not a cell. Why it's not a cell like a bacteria or any other animal or plant cell? It's because it lacks a cell membrane and a cell wall, which is the characteristic feature of any living cell. Now, since it lacks this, we call it as a particle. Another important reason is this particular particle we call as virus, which is a disease causing agent, which is an infectious agent. It is not showing any characteristics of life when it is outside the body. It is like any other object, a non-living thing. But when it gets into the body of a host cell, it could be a plant, animal, a bacteria, a living cell, then it shows the characteristics of life. Then it starts, showing living characters like, multiplication or reproduction, which is a basic character of any living organism, at the cost of the host cell. Okay, so now looking at the structure of the virus, a virus, unlike the other living cells, as I said, having a cell wall and a cell membrane, here you notice that it has a protein coat, what is called as a capsid. And you also notice that the viruses may also have an outer covering called the envelope. Apart from the capsid, they may also have an outer covering called envelope. In case they have this lipid envelope, we call such viruses as enveloped viruses. In case they have only a capsid as an outer covering and don't have this envelope, we call it as a non-enveloped virus. A coronavirus is an enveloped virus. A poliovirus is a non-enveloped virus. So you know the difference now. And at the same time, you also find proteins, viral proteins embedded in this envelope. What do they help in? They help the virus to attach to the living host cell and then enter the host cell. Why is it entering in? To multiply, to reproduce. Okay, so that's the basic characteristic of a virus. Now, does it have a genome? Like we have DNA in our cells which controls the activities of a cell. Does this particular virus have a genome or a chromosomal material? Yes, it does have a genome in the form of DNA or RNA. Our coronavirus is a RNA virus. Similarly, you have viruses which also have DNA as a genetic material based on which we classify them as DNA viruses and RNA viruses, etc. Okay, let's move on. Now that you know what a virus is and what is its basic characteristic feature, let's move on to the next slide. Coronaviruses. Coronaviruses are a group of related viruses that cause the disease in birds and mammals. Mainly they cause respiratory diseases, and these viruses are called coronaviruses because we have these as you notice this protein which is found in the envelope radiating like spikes from the envelope, which makes it look like a garland around it or a halo around it or a corona around it, and that's the reason it's called as a coronavirus. All right, now you notice the picture here, how exactly these proteins are found on the envelope, making it look like a corona around it, and hence it's called as a coronavirus. Now, when were these coronaviruses first discovered? They are the category of viruses which were discovered first in the year 1930. 1930, 1931, that was the time these viruses were studied. And they were first seen causing infectious bronchitis disease in case of chicken in North Dakota, USA. So they also called it as infectious bronchitis virus. Later on, when other similar viruses were found and they all had a similar basic structure, they were also called as coronaviruses. Now moving on to human coronaviruses. Human coronaviruses were discovered in 1960s. And which were these viruses? There were 4 different human coronaviruses that were discovered after 1960s. These are the coronaviruses which vary slightly in their protein structure, which is present in the lipid layer, and these were known to cause are known to cause common cold, just like the rhinoviruses. The viruses which cause common cold. There are 200 different types of viruses which cause common cold. And they are restricted to the upper respiratory tract, that is the nose, the throat, and may invade the sinuses. And the symptoms of common cold. They are milder, right? And you get okay with some time. And you can only treat the symptoms. You don't have any kind of medication specifically meant for getting rid of the virus, right? So exactly like that, these coronaviruses which were found after 1960s, human coronaviruses, they were causing very mild infections like that of common cold, and 15% of the common cold was is said to be caused by this coronavirus, and rest of it is by the rhinoviruses. Okay, but in the year 2002, a new coronavirus came into existence. They called it as a novel coronavirus. It was different from the earlier 4 that were discovered. This was the severe acute respiratory syndrome coronavirus, the SARS-CoV-1, which was found in 2002. And this particular virus infected several people. The virulence was not very strong as that of SARS-CoV-2, and the death rate was around 10% of the infected people, and it was well contained. Then in 2012 came the Middle East respiratory syndrome coronavirus called MERS-CoV, and this virus was also equally virulent as that of SARS, but since it was found in the Middle East countries, we called it as Middle East respiratory syndrome coronavirus. Okay, and then in 2019, you see the most severe ones among these coronaviruses, severe acute respiratory syndrome coronavirus, SARS-CoV-2. Now, why is it called SARS-CoV-2? Because its basic structure is similar to SARS-CoV-1. The symptoms caused are similar to SARS-CoV-1. Only thing, it's more infective, more virulent, easily it can be transmitted, and at the same time, you notice that there are certain glycoproteins which are different from that of SARS-CoV-1. That's why they gave it a different name called SARS-CoV-2. That causes COVID-19. More about COVID-19. How it spreads, and as I mentioned, it came from China, Wuhan, and then it started from there. And the symptoms, which include sore throat and high fever, continuous cough for different episode of 3 to 4 episodes of continuous 1 hour to 2 hours cough. And finally, many people, 85% of the people, if you have a strong immune system, you will get cured on your own. There's no medication available for COVID-19. You get okay on your own. Only thing, you may be spreading that disease to somebody else whose immune system may not be as strong as you, and sometimes you may not even show the symptoms, and you may be carrier of this virus. So no, that's one of the important features of this virus. And hand washing and maintaining physical distance, avoiding contact with these people, isolation, quarantine, a lot of things you're already aware of. Now, I would like to stress on the origin of the human coronavirus. Now, if you look at the origin of this human coronavirus, now you notice that this human coronavirus disease is a zoonotic disease. Now, what exactly is a zoonotic disease? A zoonotic disease is something that starts with the animals and then jumps to the humans. Okay, that virus which is found in animals. Animals are reservoir. It is not causing any kind of disease in them. But what is happening? That particular virus, because of close contact with humans, probably because of deforestation, wherein there's a lot of contact between humans and the wildlife, or because they are consumed, those wild animals are consumed as food, or like in Wuhan, they are sold in the wet market, and they are known for medicinal properties there, and also they serve as food. Okay, this is very common. So when there's interaction between humans and these wild animals, there's every possibility that they jump from animals to humans, like Ebola or AIDS. Or Zika virus. It's the same. But in case of human coronaviruses, you notice that the reservoir is a bat. And since it came from the bat to humans, now we call that as a zoonotic disease. A zoonotic disease, as I mentioned earlier, is a disease that the reservoir is an animal, and because of the close interaction with humans, it jumps to the humans, undergoes mutation, and causes disease in humans. Okay, now bats are the major reservoirs. Don't think this horseshoe bat, which is said to be the reservoir of our coronaviruses, where SARS-1 or SARS-2, COVID-2, they're the only ones. No, bats across the world harbor these coronaviruses. 99% of them are harmless. But in case you closely interact with these animals, in that case, there's possibility that that virus might undergo mutation and cause and use human as cell as a host cell and cause disease in host, that is humans. Okay, the same thing happened in case of a Middle East for respiratory syndrome. From the bats, it came to camels and then to the humans in the Middle East. And there could be intermediary organisms involved. Like in case of camel, it's from the bat to camel to humans. Okay. At the same time, in case of SARS-CoV-1, which came up in 2002, this was from bats to the Chinese civet, which is also sold in the wet market for its food, and from there it must have jumped to the humans. In case of SARS-CoV-2, which is seen now as COVID-19, they say from the bats, it could have come to pangolins. Pangolins, which are again sold for their scales and for their meat in Chinese wet market. So, because of close interaction with these two animals, the virus has got into the humans and has caused disease. So you understand their origin. The horseshoe bat is a reservoir through intermediary organisms. It actually passes it on to humans. That's basically because of their close interaction. All right. This has been the history now. In future, we do not know, what viruses might emerge from these animals, because of these close interactions that are still going on. Okay. So let's move on to our immune response. It's very interesting. You need to understand that whenever a disease-causing organism gets into our body shows certain kind of response. What is that called? Immune response. Why? Because our body does not want to suffer from a disease. It wants to fight the disease-causing organism. Okay, and so we have our body cells called WBCs and other proteins and chemicals and different ways in which our body fights against the disease. So we call this as an immune response. I would like to just concentrate on what happens when a virus gets into a body. Does the body simply accept it? No, it doesn't. When a virus enters into your host cell, I'm talking about a virus. When a virus enters into your host cell, the infected body cell, okay, that is here in case of COVID-2, it's the lining of the nose, lining of the eye, or it could be lining of the mouth, the mucosa lining. The cells, there, when it gets into your body, what happens is that these cells, which are infected by virus now, they start secreting certain proteins. And we call these proteins as what? Interferons. Interferons are called so because they interfere with viral multiplication. What do? They are like messengers. They go to the neighboring cells. Maybe some of the cells are infected with the virus. Some of them may be healthy cells. They go to the infected cells and give the message. Look, there is a viral infection and that's why I have been sent by my cell, and prepare yourself for viral infection. So what does a healthy cell do? A healthy cell would definitely secretes certain proteins within itself. So it knows even if the virus comes, I am not going to allow the virus to multiply. I will contain it within me so that the other cells of our body, WBCs and other lymphocytes, can come and kill me along with the virus inside. So look at that. That's how a cell responds. And these antiviral proteins, what do we call them? Interferons. Okay. Now, uh, when at the same time, you also know that the infected the cells of a body which are infected by viruses show certain changes on their surface compared to the normal cells. So, an active immune cell like a T cell or a B cell we call in our body. Now, those cells will come and respond to this viral infection. In what way? Uh, they may go and engulf the entire virus infected cell, or they may secrete certain poisons onto them, cytolysins we say, and kill that cell along with the virus inside. Or they may also secrete something called as antibodies. What are antibodies? Y-shaped proteins. Who secretes them? Our immune cells. Why is it secreting it? It's a defensive protein. It can go and attack the virus and contain the spread of viral infection, right? So these proteins which are secreted by our body uh, are called antibodies in order to combat a infection. You get that? Keep this in mind. Remember this. These are the proteins which are defensive, which are secreted by our body, by our immune cells. Whenever a pathogen comes into our body, here it's against the virus that has invaded. Now you all know what a vaccination is. Vaccination is a process of injecting a vaccine or giving a vaccine. A vaccine is a biological preparation that provides immunity to a particular infectious disease. Now, many countries are trying to find a vaccine for COVID-19. Uh, and they will be successful definitely in a couple of months. Now, a vaccine cannot be simply given to humans because it may cause some side effects later. And you know it contains a dead virus or a weakened virus or any toxins of the virus, which can actually, evoke this response in our body. As I mentioned, when this virus which is weakened or which is dead or which some of its chemicals are injected in our body in the form of a vaccine, our body also recognizes it and responds to it by secreting what? By secreting antibodies. So this is natural immunity that you're getting, right? And these antibodies will remain in our body for, in case of COVID-19, they say 1 or 2 years, and or a SARS-1, they have found that it remains for 1 or 2 years. Similar thing maybe with COVID-19, and then they finally die out. But at least 1 or 2 years, you have that protective protein in your blood. Same way in vaccination, they're trying to do it artificially by injecting a weakened virus into your body, which will not be able to cause any symptom or disease, but at least will be able to give you protection by helping your body secrete antibodies. We call it as artificial immunity. Alright, now moving on to the testing. How is the testing done in case of COVID-19? In COVID-19, there are two ways in which the testing is done. If you have symptoms of they want you, if you go for a test for COVID-19, one is they take a oropharyngeal swab, or they may even take a nasopharyngeal swab. It's nothing but from the deep inside your throat and deep inside your nose where the virus harbors itself. So these uh, swabs are sent to the laboratory, and then they check and tell you whether you have the virus in your body or not. So that's positive or negative. And if the viral concentration is very less in the beginning of the disease, you may even test negative. But after some time, when the symptoms worsen, you go and test again, you may be positive. That depends on the concentration of the virus that you harbor. So you must have heard of cases where initially the person tested negative, but later he tested positive. It's not the problem with the lab. It's a problem with the concentration of the virus. Okay, and then that's about testing for the virus. Now, what are serological tests then? Serological tests are where it's a blood test, okay, where the blood of a person is taken, and tested with the help of a test kit for COVID-19. What are they checking actually in your blood? If you have antibodies. Okay, that is a serological test. Now, when will you have antibodies? In case you have been infected earlier, your body would have reacted and produced antibodies. So, when they check with this COVID-19 kit, okay, when they do a blood test on you, they come to know whether you've been infected earlier or not, right? So, if you have antibodies, you are infected. If you're not having antibodies, that means you are still prone to a disease, you're not infected. And there is no scientific evidence that once you get the disease, you are not supposed to get the disease the second time in case of COVID-19. Okay, it's not like measles and other disease, chicken pox and all that, where you get it once, for several more years, you won't get it, you have immunity. COVID-19, there's not much of scientific evidence for anything because hardly the virus for a few months it's been there, and there's not much of testing done on this virus, and we don't know much about this virus in fact, and with time they will definitely find out. And now moving on to the end of this session, one thing you should remember is not to panic, to always keep calm, okay, and in case you stay home and stay safe, lockdown period is for that, so that you don't spread the disease or you don't contact the disease. At the same time, World Health Organization's website and Government of India's website will give you exactly information, authentic information. Don't go for WhatsApp messages and what you get to hear from people about this disease. Okay, stay home and stay safe. Thank you for listening. Hope you have got some information that of importance, something that you learned from this particular slide. Thank you again.